Welcome to the Deep Dive. We're here to give you that shortcut, that deep structural understanding of complex topics. And today, wow, we're really plunging headfirst into the pathophysiology of infection.
We are, and it's a field that, it's built on a pretty relentless principle.
Yeah.
Every living thing, microbes included, is just trying to survive and reproduce.
Right.
And for countless microorganisms, their environment, well, it's us. It's the intricate ecosystem of the human body.
And that's where the conflict starts, isn't it? When one of these pathogenic organisms manages to get past our defenses, skin, mucous membranes, the whole immune setup.
Exactly. If those defenses fail and the organism starts causing injury, that's when you get infectious disease.
We're not just talking symptoms here. It feels more like a fundamental disruption of biological balance.
It really is. So our mission today, if you like, is to weave together three key disciplines: microbiology, immunology, and epidemiology. We want to give you a framework, basically, to clearly connect the dots between how these diseases work, the mechanisms, and what you actually see clinically, the outcomes in patients.
Okay, let's unpack this. A good place to start seems to be that foundational idea.
Yeah.
The triad of infectious disease.
Yeah.
Agent, host, environment.
That's the core. But, before we dig deeper, we need some clear language. Let's define the terms of engagement, so to speak.
Good idea.
First, the host. That's simply any organism providing the nutritional and physical support another organism needs to grow.
Straightforward now.
But then there's a crucial difference between colonization and infection. Colonization just means establishing a presence, like bacteria setting up shop on your skin.
Okay, so they're there, but not necessarily causing trouble.
Right. Infection is when they're not only present, but they're multiplying and causing injury to the host. That injury part is key. No injury, technically no infectious disease.
And, surprisingly often perhaps, that interaction isn't harmful at all, is it? It exists on a spectrum.
Absolutely. On one end, you have commensalism. The microbe gets something from us, say nutrition, but we, the host, are completely unharmed. A lot of our normal microflora fit here.
Just hanging out.
Pretty much. Then you've got mutualism, which is a genuine win-win. Think about some bacteria in our gut.
The vitamin producers.
Exactly. They get a stable environment and nutrients, and in return, they make essential things like vitamin K, which we then absorb and use.
That's such a great clinical link. It immediately clicks why newborns who, haven't built up that gut flora yet need that vitamin K shot. They're missing their mutualistic partners.
Precisely. It's applied pathophysiology right there. Now, the other end of the spectrum, that's the parasitic relationship.
Where only the microbe wins.
Right. The infecting organism benefits and the host gets injured. The organisms that do this, we call them pathogens, and their capacity to cause that disease, that's their virulence.
So most things we bump into are harmless saprophytes just living freely in the environment. But the line can blur, can it? When can our own friendly flora turn nasty?
That's the concept of opportunistic pathogens. It happens when the host's defenses are down, maybe due to illness, malnutrition, certain medications like chemotherapy. The normal flora, which are usually kept in check, seize the opportunity of that weakened immunity. They overgrow, invade, and cause disease they normally wouldn't.
Okay, let's shift focus to the agents themselves. You mentioned 6 main categories: prions, viruses, bacteria. What else?
Right. Prions, viruses, bacteria, then some hybrid agents, Rickettsiaceae and Chlamydiaceae fungi, and parasites. A real rogues gallery.
Let's start with prions. These are just mind-bending, infectious proteins. No DNA or RNA involved.
It really challenges everything we thought we knew about infectious agents. They're essentially misfolded proteins.
So how does a protein, just a protein, cause devastating neurodegenerative diseases like CJD or mad cow disease?
It's a process of, structural corruption almost.
Huh?
A normal cellular protein somehow gets misfolded into this abnormal protease-resistant form. We call it PrPSC. This rogue PrPSC then acts like a template. It bumps into normal proteins and forces them to misfold into the abnormal shape too. They clump together, aggregate, and cause progressive damage and death of neurons.
Terrifying. And because they aren't really alive in the traditional sense.
Exactly. No metabolism, no reproduction in the usual way. So antimicrobials, completely ineffective against them.
Right. Okay, next up, viruses. The smallest pathogens. Basically a protein code that caps it around a core of genetic material, DNA or RNA, but never both.
And that structure dictates their lifestyle. They're obligate intracellular pathogens. They absolutely have to get inside our cells and use our cellular machinery to replicate. They can't do it on their own.
And the results vary hugely. Some just replicate and burst the cell, but others are sneakier, right? Like herpes viruses.
Yeah, some establish latency. They insert their genetic material right into our own host chromosomes and just wait. They can stay dormant for years, even decades. That's why chickenpox, caused by a herpes virus, can reappear years later as shingles.
Wow. And then there are retroviruses, like HIV. They have that special trick.
They do. They carry an enzyme called reverse transcriptase. It allows them to do something very unusual, transcribe their RNA genome back into DNA.
The reverse of the usual flow of genetic information.
Exactly. And that DNA copy can then be integrated permanently into the host cell's genome.
And we also have viruses linked to cancer, oncogenic viruses.
Yes. Some viruses have the ability to actually transform normal host cells into malignant cancerous cells. HPV, human papillomavirus, is a major example, strongly linked to cervical cancer.
Okay, moving on to bacteria. These are different prokaryotes, single-celled, replicating on their own, no organized nucleus.
Correct. And we classify them in a few key ways. By shape, cocci are spheres, bacilli are rods, spirilla are spirals, and crucially, by how they react to the Gram stain, which tells us about their cell wall.
That Gram stain is so fundamental in the clinic. Gram-positive bacteria stain, purple thick peptidoglycan layer. Gram-negative stain, red thin peptidoglycan, plus that outer membrane.
That outer membrane containing lipopolysaccharide, LPS, which, as we'll discuss, is a potent endotoxin.
Right. But bacteria don't just float around solo, do they? They have survival strategies, biofilms.
Oh yeah, biofilms are huge. Bacteria often live in these structured cooperative communities. They stick to surfaces, embedded in a kind of slime matrix they produce.
And they talk to each other, quorum sensing.
They do. They use chemical signals to coordinate their behavior, things like gene expression, toxin production, defense against antibiotics. It makes biofilms incredibly resistant.
And some bacteria have unique structures for movement, like Spirochetes, thinking about Lyme disease.
Yes, Borrelia burgdorferi, the Lyme agent, is a classic spirochete. It has this corkscrew shape and internal flagella that allow it to literally drill through tissues and spread throughout the body.
Like in that case study, Mrs. Roon. Her initial tick bite was localized, but the bacteria spread, causing systemic issues, headache, fatigue, joint pain.
Exactly. It highlights the importance of motility for dissemination. And then you have bacteria at the other extreme, like mycoplasmas.
The tiny ones.
The smallest free-living bacteria. And their key feature, no rigid cell wall at all.
Which sounds like a weakness, but
It's actually a strength against certain drugs. Antibiotics like penicillin work by targeting cell wall synthesis. If you don't have a cell wall, penicillin can't hurt you.
clever. Okay, what about those hybrid agents? Rickettsiaceae and Chlamydiaceae, sort of virus-like, sort of bacteria-like?
They occupy a middle ground, yeah. Like viruses, they're obligate intracellular. They have to live inside host cells. But like bacteria, they reproduce asexually, have cell walls, though sometimes atypical, and contain both DNA and RNA.
And how do they spread?
It differs. Ricketsiae, causing diseases like Rocky Mountain spotted fever, are typically transmitted by arthropod vectors, ticks, lice, fleas. Chlamydiaceae, often causing STIs or eye infections, are usually spread through direct person-to-person contact.
Got it. Now, fungi. Eukaryotes, like us, but mostly living as saprophytes, yeasts and molds.
That's the basic split. Yeasts are single-celled, usually reproduced by budding. Molds are filamentous, forming networks of hyphae called a mycelium.
And where they cause infection often comes down to temperature.
That's a key factor, yes. Think about superficial mycoses, like athlete's foot or ringworm. The fungi causing those, dermatophytes, generally can't grow well at our core body temperature, 37 degrees Celsius.
Ah, so they stick to the cooler skin surfaces.
Exactly. Whereas organisms causing systemic fungal infections, like Candida albicans, can grow happily at 37 degrees. That allows them to invade deeper tissues and potentially cause life-threatening disease, especially in immunocompromised hosts.
Makes sense. And finally, parasites, actual members of the animal kingdom.
Yep. We group them loosely. There are the unicellular protozoa, think malaria, giardia, then the helminths, which are worms, tapeworms, roundworms, flukes. You often get those through ingestion or sometimes skin penetration.
And arthropods, ticks, lights.
Right. Arthropods can act as vectors, transmitting other pathogens like we saw with Rickettsiaceae and Lyme disease. Or they can be ectoparasites themselves, living on the host, like scabies, mites, or lice.
Okay, quite the lineup of potential troublemakers. Let's shift gears slightly to epidemiology, how we track these diseases in populations. Incidence and prevalence.
Two fundamental measures. Incidence is the rate of new cases occurring in a population over a specific time period. Prevalence is the total number of active cases, both old and new, present at a particular point in time.
And the patterns tell us about the threat level: endemic, epidemic, pandemic.
Exactly. Endemic means a disease is found at a relatively stable expected rate within a geographic area. Think seasonal flu, usually.
Okay.
An epidemic is when there's an abrupt, unexpected increase in the incidence of a disease, well above the endemic rate.
Like a sudden outbreak.
Right. And a pandemic is essentially an epidemic that spreads across continents, sometimes worldwide. The SARS coronavirus outbreak you mentioned earlier started as an epidemic but quickly became pandemic due to global travel.
It shows how interconnected we are. Where do these infections actually come from? Endogenous versus exogenous.
Good distinction. Endogenous infections arise from the host's own microbial flora, think opportunistic infections. Exogenous infections come from the external environment, water, food, soil, other people, animals.
And objects can spread them too. Fomites.
Yes, fomites are inanimate objects that get contaminated and transmit pathogens. Doorknobs, keyboards, toys in a daycare spreading cold viruses.
Ugh. And zoonoses. Animal to human.
Right, zoonoses are diseases naturally transmitted from animals to humans. Lyme disease from deer ticks is a perfect example. Rabies from animal bites, lots of them. We also distinguish nosocomial or healthcare-associated infections from community-acquired ones.
So how do these agents actually get in? The portal of entry.
Multiple routes. Penetration involves any break in the skin or mucous membranes. Could be an injury, surgery, an IV line, even an insect bite.
Direct contact.
Yep. Direct contact with infected tissue or secretions. Many STIs spread this way. Also includes vertical transmission from mother to child during pregnancy or birth, leading to congenital infections. Think about the torch infections, toxoplasmosis, other, rubella, cytomegalovirus, herpes.
Okay. Ingestion. Eating or drinking contaminated stuff.
Ingestion via the mouth and GI tract. But the pathogen has to be tough. It needs to survive stomach acid, digestive enzymes, peristalsis.
Some must be better at that than others.
Definitely. Shigella, for instance, which causes severe dysentery, is very acid resistant. It means a very small number of bacteria, a low infectious dose, is enough to make you sick. Others need a much larger dose.
And inhalation. Breathing them in.
Inhalation is very efficient for respiratory pathogens. They have to get past the defenses in our airways, mucus, cilia. Things like streptococcus pneumonia, influenza virus, measles virus spread this way.
Okay, so the pathogen is in. What does the course of the illness actually look like for the patient?
There are stages, right? Yes, a typical acute infection follows a predictable pattern, though the timing varies wildly. It starts with the incubation period.
Pathogens multiplying, but you feel fine.
Exactly. No symptoms yet, but the organism is establishing itself. Then comes the prodromal stage. This is when you start feeling off. Vague symptoms like malaise, maybe a low fever, muscle aches. Not specific yet.
Kind of feel like you're coming down with something.
That's the one. Then it ramps up to the acute stage. This is peak illness. Symptoms are most severe, most pronounced, and usually specific to the disease, allowing for diagnosis.
That's when you know you're really sick.
Right. If the host defenses start winning, you enter the convalescent period, the infection is being contained, damage is being repaired, symptoms start to subside.
Feeling better, finally.
And ideally, it ends in the resolution stage. The pathogen is totally eliminated from the body, health is restored.
But it doesn't always follow that neat path, does it?
Yeah.
Chronic infections.
No, there are variations. Chronic disease can linger for months or years, sometimes with flare-ups. Fulminant illness is the opposite, incredibly rapid, severe onset, often with little or no prodrome. Some infections are subclinical, causing no noticeable symptoms at all.
And how do we talk about where the infection is? You hear acunus and axemia a lot.
Standard medical terminology. The suffix "-itis" just means inflammation of a specific site. Appendicitis, meningitis, dermatitis.
Inflammation of the appendix, meninges, skin. Got it.
And ischemia means the presence of something in the blood. Bacteremia is bacteria in the blood. Viremia is viruses in the blood.
And that relates to sepsis, which has a new definition now.
It does, and it's crucial. Sepsis isn't just infection plus inflammation anymore. The current definition emphasizes a dysregulated host response to infection.
Meaning the body's reaction is out of control.
Precisely. It's the body's over-the-top response that causes life-threatening organ dysfunction. It recognizes that often the host's reaction is doing as much or more damage than the pathogen itself.
That's a major shift in thinking. And we distinguish localized from systemic infections.
Yes. Localized means confined to a specific area, like a skin abscess. Systemic means it has spread throughout the body, often via the bloodstream or lymphatics. Mrs. Rune's Lyme disease became systemic.
And what makes one pathogen more dangerous, more virulent than another? They have specific weapons.
They do. We call them virulence factors. These are the molecules or structures the pathogen uses to cause disease. We can group them into, say, 4 main categories. First, toxins.
Okay, poisons, basically.
Essentially, exotoxins are proteins actively secreted by bacteria, often gram-positives, but sometimes gram-negatives too. They can be incredibly potent and specific. Think neurotoxins, botulinum toxin causing flaccid paralysis, botulism, tetanus toxin causing spastic paralysis, tetanus.
Scary stuff.
Very. Some exotoxins act as superantigens. Toxic shock syndrome toxin, TSST1, from Staphylococcus aureus is a classic example.
What makes it super?
Instead of activating just a few specific T cells, it non-specifically links immune cells together, triggering a massive, uncontrolled release of inflammatory cytokines. Basically, it sends the immune system into overdrive, leading to shock.
Okay, that's exotoxins. What about endotoxins?
Endotoxin is different. It's not actively secreted. It's a structural part of the outer membrane of gram-negative bacteria, that lipopolysaccharide LPS we mentioned.
So it only gets released when the bacteria die and break apart.
Primarily, yes. Even tiny amounts of LPS are incredibly potent inflammatory signals. It activates immune cells, the complement system, the clotting cascade. It can cause fever, low blood pressure, disseminated intravascular coagulation, DIC, and ultimately potentially fatal endotoxic shock.
Right. So toxins are one weapon. What else? Adhesion.
Absolutely critical. Adhesion factors are molecules on the pathogen surface, ligands or adhesins that bind specifically to receptors on host cells. If a pathogen can't stick, it usually gets washed away, it can't colonize, can't invade.
Makes sense. Stickiness matters. Then evasion. Hiding from the immune system.
Huge category. Evasive factors help the pathogen avoid being detected or destroyed. Bacterial capsules are a classic example. That slippery outer layer prevents phagocytic cells from grabbing and engulfing them.
What else?
Some bacteria produce enzymes that neutralize immune components. Others, like Borrelia causing Lyme, use antigenic variation. They constantly change their surface proteins, so by the time the immune system mounts a response to one version, the bacteria have switched to another. It's like changing disguises.
Constantly moving target. And the last category. Invasion.
Invasive factors are usually enzymes that help the pathogen break down host tissues and spread. Things like collagenesis that degrade collagen, hyaluronidase that breaks down hyaluronic acid, the glue between cells. They basically clear a path for invasion.
Okay, so we have the agents, the mechanisms, the disease course. How do we actually diagnose these things in the lab?
Diagnosis always hinges on 2 things: compatible clinical signs and symptoms in the patient, plus evidence of the presence of the suspected pathogen. Lab techniques give us that evidence.
Like growing it in a dish. Culture.
Culture is the gold standard for many bacteria and fungi. You take a sample, blood, urine, sputum, and try to grow the organism on specific nutrient media. If it grows, you can identify it, test its antibiotic susceptibility.
But that doesn't work for everything. Viruses.
Right. Obligate intracellular pathogens like viruses, Rickettsiaceae, Chlamydia won't grow on artificial media. They need living host cells. So you use cell culture techniques, you infect cultured cells and look for the cytopathic effect, CPE, visible changes or damage to the cells caused by the virus.
Okay. What if you can't easily culture it? Serology.
Serology is an indirect method. Instead of looking for the pathogen itself, you look for the host's immune response to it specifically, antibodies in the patient's serum.
IgM and IgG.
Exactly. A rise in IgM antibodies typically indicates a recent acute infection. IgG antibodies rise later and often persist long-term, indicating past infection or immunity. It's very useful for viruses like hepatitis B or diagnosing congenital infections.
And the really high-tech stuff, DNA and RNA methods.
Hugely important now. DNA probe hybridization, and especially polymerase chain reaction, PCR.
PCR is like a molecular photocopier, right?
That's a great analogy. PCR uses specific DNA primers and cycles of heating and cooling to find even tiny amounts of a pathogen's unique genetic material, DNA or RNA, in a sample and amplify it exponentially. Make millions or billions of copies.
So you can detect it even if there's very little there.
Precisely. It's incredibly sensitive and specific. And real-time PCR is even faster and allows quantification. We use it for rapid diagnosis of things like C. difficile toxin genes, or, crucially, for monitoring viral load in patients with HIV or hepatitis C.
To see if the treatment is working by reducing the amount of virus.
Exactly. It guides therapy.
Okay, diagnosis sorted. What about treatment?
Broadly, three approaches: medical, antimicrobials, immunologic, and sometimes surgical.
Antimicrobials, antibiotics, antivirals.
Right. Antibacterials, antibiotics target specific bacterial processes, cell wall synthesis, like penicillin, protein synthesis, DNA replication. Antivirals are trickier because viruses use our own cell machinery.
So you have to target something unique to the virus.
Ideally, yes. A cycle V targets viral DNA polymerase. HIV drugs often target unique viral enzymes like reverse transcriptase or protease. Antifungals frequently target ergosterol, a sterol found in fungal cell membranes, but not human ones.
But the big shadow looming over antibiotics is resistance.
It's arguably the biggest public health crisis we face. Bacteria evolved defenses, rapidly producing enzymes like beta-lactamases that chew up penicillin-like drugs, altering the target site, pumping the drug out.
And misuse of antibiotics drives this.
Absolutely. Every time we use an antibiotic, especially inappropriately, we create selective pressure favoring the resistant strains. Antibiotic stewardship, using these precious drugs wisely, only when needed, for the right duration, is critical. We're running out of options.
A sobering thought. What about immunotherapy?
Using the immune system itself can involve giving intravenous immunoglobulin, IVIG, pooled antibodies from donors to provide passive immunity, or using cytokines like interferons to stimulate the patient's own immune response. And of course, vaccination is preventative immunotherapy.
Yeah. And surgery. When is that needed?
Sometimes it's essential. Draining an abscess to remove the pus and bacteria, debriding dead tissue, removing an infected medical device like a heart valve, or joint replacement biofilms on implants are often impossible to clear with antibiotics alone.
Right, the physical removal is necessary. Okay, wrapping up, we have to touch on, well, the darker side. Bioterrorism.
Unfortunately, yes. Certain infectious agents have potential for use as weapons. Public health agencies categorize them based on risk, ease of spread, mortality rate. Category A agents are the highest risk.
Like anthrax, smallpox.
Exactly. Bacillus anthracis, anthrax, variola virus, smallpox, Yersinia pestis, plague, botulinum toxin. These require special preparedness and response networks, like the Laboratory Response Network, LRN.
And beyond intentional threats, there's a constant emergence of new global diseases.
Globalization makes it inevitable. International travel, global trade, pathogens move easily. We saw West Nile virus emerge in the U.S. in 1999, carried by mosquitoes. SARS coronavirus spread rapidly via air travel in 2003.
And Zika.
Zika virus, another mosquito-borne flavivirus, spread through the Americas, causing concern due to its link with congenital microcephaly in babies born to infected mothers. It really highlights how a localized outbreak can become a global health emergency very quickly.
So pulling this all together, what's the main takeaway? It feels incredibly complex.
It is complex. The outcome of any encounter between a microbe and a host depends on so many variables. The inherent virulence of the organism, how many organisms you're exposed to, the infectious dose, the portal of entry, but maybe most importantly, the state of the host's defenses. Your health, your immune status, your genetics, it all plays a role.
That's a dynamic battle every time.
It really is. And maybe a final provocative thought to leave you with. We're getting better at treating the acute phases of severe infections, like sepsis. Survival rates are improving.
That's good news.
It is, but the story doesn't end there. We're increasingly recognizing that the pathophysiology extends far beyond the initial recovery. Survivors of critical infections often face long-term consequences: chronic pain, fatigue, significant cognitive impairment, functional disabilities, higher risk of later cardiovascular events.
Wow. So surviving the infection is just the first hurdle. The long-term impact can be profound.
Exactly. The true burden of infectious disease often stretches out for years, impacting quality of life long after the pathogen is gone. It underscores why understanding these mechanisms so deeply is crucial, not just for acute care, but for long-term patient well-being.
That is a really critical insight, a powerful place to end. We certainly hope this deep dive into the mechanisms of infectious disease has been a valuable resource for you. Thank you so much for joining us for this essential discussion.
And indeed, a warm thank you from the Last Minute Lecture team.